Hello friends, in this video now I'm going to take this topic Krebs cycle or TCA cycle. First we'll be understanding the names. One name is TCA. Why the name TCA? The full form is tricarboxylic acid. Why the name tricarboxylic? Tricarboxylic means 3 COOH group, 3 carboxy group present in some compound. And let me tell you, the first two compounds formed in TCA, that is citrate and isocitrate, they are tricarboxylic acids, right? Now the other name is Krebs cycle by the name of the scientist. So Krebs cycle if written means TCA cycle. If Krebs-Hanslet cycle written, be careful, if Krebs-Hanslet cycle written, that is not TCA cycle, that is urea cycle, right? Now there's one more name of TCA cycle, that is citric acid cycle, because the first compound formed in citric acid cycle is citrate. That's why the name citric acid cycle, because the first compound formed is citrate. Now, what is the difference between citric acid and citrate? Citric acid is COOH, acid group, COH group. Citrate means the ionized form, that is COO minus, right? In body, everything is ionized. So in body, it is present as citrate, right? Now TCA cycle, tell me, it is anabolic or catabolic pathway? It is both anabolic as well as catabolic, and therefore it is known as amphibolic pathway. TCA is amphibolic pathway. Like both polar, non-polar known as amphipathic, similarly both anabolic, catabolic is known as amphibolic. When I say TCA is catabolic, what that means? What is the catabolic role of TCA? Catabolic role of TCA means all the macromolecules, carbohydrates, fats and proteins, they all, when they are getting broken down in body, they all form acetyl-CoA, which enters TCA cycle. So can we say that all macromolecules are getting broken down and they finally reach TCA cycle? So TCA cycle is the common catabolic pathway, right, of all the macromolecules, because all the macromolecules gets broken down and reach TCA cycle. Now what is anabolic role of TCA? Anabolic role of TCA, anabolic means synthesis, because many TCA intermediates are used for the synthesis of important compounds in the body. TCA intermediates are used for synthesis of many compounds in the body. For example, succinyl-CoA is intermediate of TCA cycle, which is used for the formation of heme in the body. If you remember, the first step of heme synthesis is succinyl-CoA plus glycine makes ALA, that is delta aminolevulinic acid, right? So intermediate of TCA cycle is used for the synthesis of some important compound in the body. So therefore this is anabolic role of TCA cycle, right? Also TCA cycle is a vital cycle for the cells. Vital cycle, because it is a vital cycle, it always has to occur. Therefore there is no enzyme deficiency of TCA cycle. This kind of topic is not there in your books. Enzyme deficiency of TCA, not there, because it is a vital cycle. It always has to occur. Like there's no life without TCA cycle, right? See, on one hand, I'm saying TCA cycle is vital cycle. That means all the TCA intermediates should always be present in any cell of the body. They should be maintained at constant level. And on the other hand, I'm saying that anabolic role of TCA, those reactions are using TCA intermediates. So that means there must be some reactions which are producing TCA intermediates, because anabolic role of TCA is using TCA intermediates. So there are some reactions which produce or which replenish TCA intermediates. Those are known as anaplerotic reactions. So anaplerotic reactions are those reactions which replenish TCA intermediates. For example, pyruvate can form oxaloacetate. OAA when I write, that is the short form of oxaloacetate. See how this reaction will occur. Pyruvate is a 3-carbon compound, oxaloacetate is a 4-carbon compound. So from 3 to 4 carbon, we can simply add carbon dioxide, and the name of this enzyme is pyruvate carboxylase. So pyruvate carboxylase, which is synthesizing oxaloacetate. This is the most important anaplerotic reaction. Now solve this question. TCA is activated by which hormone? Insulin, glucagon, both or none? I told you TCA is amphibolic, both catabolic, anabolic. You know anabolic pathways are activated by insulin, catabolic pathways are activated by glucagon. So most of the students will answer both for this question. But answer is not both, answer is none. Because TCA is not having any hormonal control because it is a vital cycle of the body. It does not depend upon hormones. See, when I say TCA is activated by insulin, it would mean that when we are in fed state, when we are eating food, then TCA will occur. And if I say it is activated by glucagon, then it would mean that TCA is occurring when we are in fasting state. But no, TCA does not depend on our food status. It is a vital cycle for the cells. It will always occur, does not depend on food status. So I will say that TCA has no hormonal control because it is a vital cycle for the cells. Now, when I say it is not controlled by hormones, so that doesn't mean TCA will just go on and on and on till infinity. No. There's some control, right? Something has to control TCA cycle. So let's see, TCA is controlled by which thing? TCA is controlled by, see, mainly TCA in the cells is for energy. So directly energy status is controlling TCA cycle. So energy status of the cell. That is, if a cell has not any ATP, the cell is not having ATP, then TCA will occur, right? But if a cell has enough ATP, then TCA will not occur, right? So simply it directly depends on energy status of the cell. And second, it depends on availability of oxaloacetate. Sometimes students say it depends on availability of acetyl-CoA, right? But no, it is not acetyl-CoA. It depends on the availability of oxaloacetate. That's why oxaloacetate is regarded as carrier of TCA cycle. Oxaloacetate is regarded as first substrate of TCA cycle. And also oxaloacetate has catalytic role in TCA cycle. When I say catalytic role, what that means? See, enzymes have catalytic role. Enzymes are catalyst. What that means? That enzyme is not used up in the reaction. Right? Enzyme is obtained as such after the reaction. Similarly, oxaloacetate is again regenerated in TCA cycle. It is not used up in TCA cycle. See what that means? Catalytic role. Oxaloacetate will combine with acetyl-CoA as the first step of TCA cycle, making citrate. After citrate, there are so many steps. Finally, oxaloacetate is again regenerated back. So we say that this oxaloacetate is having catalytic role in TCA cycle. Because oxaloacetate has a catalytic role, therefore TCA is not a pathway. It is a cycle. See, because oxaloacetate is again obtained. So it is not a pathway. Pathway means going in one direction like glycolysis, right? Glycolysis is a pathway. Glucose forms pyruvate. But in TCA, we are starting from oxaloacetate as the first substrate. In the end also we are getting oxaloacetate. So we say that TCA is a cycle. It is not a pathway. Right? Now let's see few compounds. For example, glutarate. Tell me what is the difference between glutarate and glutamate. Glutarate, we do know alpha ketoglutarate, intermediate of TCA cycle. Glutamate is amino acid. There is one more, glutamine. So So the names are matching here. So you have to be very clear for the difference in them. The similarity is that all these three things have 5 carbons. Now it is very easy to learn the difference when I have written these 3 compounds like this. On right hand side, make arrow and keep on adding amino group. So if you add 1 amino group in alpha ketoglutarate, it becomes glutamate. You add 1 more amino group, it becomes glutamine, right? Similarly, oxaloacetate will form aspartate, will further form asparagine. Now all these compounds, they have 4 carbons, and again same, keep on adding amino group, that is add 1 amino group in oxaloacetate, it will become aspartate. Add 1 more amino group in aspartate, it will become asparagine. Now see here, can you see these 4 are amino acids? Glutamate, glutamine, aspartate, asparagine. These 4 amino acids are getting synthesized from TCA cycle intermediates. I told you this is anabolic role of TCA, that TCA intermediates are used for the synthesis of important things in the body, and here you can see 4 amino acids, obviously non-essential amino acids, they are getting formed in body from TCA intermediates. So this is anabolic role of TCA cycle. Also you have to note that succinate or succinyl-CoA, this compound is also having 4 carbons, right? Then let's see the difference between malonate. There is a compound malonate, other is malate. Malonate, when this N is there, this is a 3 carbon compound, and malate is a 4 carbon compound. Malonate is inhibitor of TCA cycle and malate is intermediate of TCA cycle, right? Now this compound malonate or malonyl-CoA, this compound, this is a 3 carbon compound, you know, it is inhibitor of 3 pathways in the body. One is TCA cycle, ETC and beta oxidation of fatty acid. Actually, it inhibits succinate dehydrogenase of TCA cycle. It will inhibit complex 2 of ETC and it inhibits CPT1 of beta oxidation of fatty acid. What is the full form of CPT? It is carnitine palmitoyl transferase 1, right? And also tell me, what is the full form of CPS enzyme? CPS is carbamoyl phosphate synthetase 1 and 2 we have. CPS1 is in urea cycle. CPS2 is in pyrimidine synthesis, right? But this I have written is CPT carnitine palmitoyl transferase 1 and 2, which are there in beta oxidation of fatty acid. Now malonate inhibits CPT1 of beta oxidation of fatty acid, right? Then note down, all the enzymes of TCA cycle are lying in mitochondrial matrix except one, and that is succinate dehydrogenase, which is lying in inner mitochondrial membrane. You know, in inner mitochondrial membrane, ETC occurs. This enzyme has also a role in ETC, that is complex 2 of ETC, that we'll be seeing later in another video for ETC, right? Now let's start with the cycle, Krebs cycle. You know the starting material or the first substrate we say, first substrate is oxaloacetate. I told you oxaloacetate is 4 carbon compound. This oxaloacetate will combine with acetyl-CoA, which is 2 carbon. They both join to make citrate. So 4 plus 2, this is 6 carbon compound. And the name of enzyme is citrate synthase. I told you citrate synthase belongs to transferase category, that is EC number 2, right? Next is citrate gets converted to its isomer, that is isocitrate. So this is also 6 carbon compound. And name of enzyme is aconitase. This aconitase is converting one isomer to another. So EC number should be 5 here, but it is not 5. Aconitase is a lyase, that is EC number 4. Also note down fumarase is also a lyase, right? Now see the next 2 steps of TCA cycle. This step and this step, that is isocitrate to alpha ketoglutarate to succinyl-CoA. These 2 steps are same like we have done in link reaction. You remember link reaction was 3 carbon to 2 carbon, removal of carbon dioxide, name of enzyme is dehydrogenase. So that was oxidative decarboxylation requiring B1, and name of enzyme is dehydrogenase here, and also NAD forms NADH, right? Next two steps of TCA are same like link reaction, that is 6-carbon isocitrate gets converted to 5-carbon alpha-ketoglutarate, then this 5-carbon gets converted to 4-carbon succinyl-CoA. So in both the steps, carbon dioxide is getting removed. Name of enzyme is dehydrogenase. The first one is isocitrate dehydrogenase. The next one is alpha-ketoglutarate dehydrogenase. In both these steps, B1 vitamin used, oxidative decarboxylation, and NAD forms NADH, right? So we have reached till this point, succinyl-CoA. Now see here, we are done with this half of TCA. In this half of TCA, we have done 2 dehydrogenases. They are doing oxidative decarboxylation. And see this, now this half of TCA, where we are starting from succinyl-CoA, and after all these reactions, we are ending at oxaloacetate. Oxaloacetate is also 4-carbon, succinyl-CoA is also 4-carbon. So all the intermediates in between these two are 4-carbon compounds. Succinate, fumarate, malate, all are 4-carbon compounds. And whenever dehydrogenase on this side of the cycle, see succinate dehydrogenase, malate dehydrogenase, there is no change in number of carbons when these two dehydrogenases are working. So they both are simply oxidation steps occurring. You know, dehydrogenase means removal of hydrogen, means oxidation, right? Now let's see the next step in detail, that is succinyl-CoA gets converted to succinate. You know that CoA is a high energy bond. Now this bond is getting broken in this step. Succinyl-CoA gets converted to succinate. So CoA is a high energy bond. When this bond is broken, energy is released. That energy can be trapped in the form of ATP or GTP. And here GTP is written, and you know ATP GTP formed at some step is known as substrate level phosphorylation step. In glycolysis, we have done 2 substrate level phosphorylation steps, but in TCA cycle, this is the only substrate level phosphorylation step occurring. And you know, enzyme must be some kinase. Here it is succinate thiokinase. So what is the EC number here? See, I told you if it is kinase, then EC number is 2. But if thiokinase is written, then EC number is 6, right? Be careful. If thiokinase is written, then EC number is 6, right? Now, in this step, whenever you see this diagram TCA cycle in any of the book, then here in this step, they write GTP formed, right? But let me tell you, this is wrong. Actually, most of the time, this enzyme will form ATP and not GTP. Most of the time, it will form ATP. If you will see your latest edition of Harper, 30th edition, in this book, they have done this correction that in the diagram, they have written ATP here because most of the time, this enzyme forms ATP. So, if they simply ask you a question, thiokinase produces what thing? Thiokinase produces what? You will say ATP or GTP? You will say ATP because I told you most of the time this enzyme will form ATP. But if they ask you thiokinase in liver, in kidney, and if they ask you during starvation, if these 3 conditions they ask you, in liver, in kidney, during starvation, it produces what? Then you will say GTP. Understand why? Because during starvation, this pathway occurs, gluconeogenesis occurs. Actually, this pathway gluconeogenesis requires GTP, and gluconeogenesis, you know, occurs in these 2 organs, liver and kidney. So, can I say that thiokinase of TCA produces GTP in liver and kidney during starvation for gluconeogenesis, right? You know, gluconeogenesis is a high energy consuming pathway. This pathway requires GTP and that is provided by thiokinase of TCA cycle, right? So we have reached till this point, succinate. Let's see the next step. Succinate to fumarate. There is no change in number of carbon. Enzyme is dehydrogenase, that is succinate dehydrogenase. So this is simple oxidation step occurring and somebody will come to take this hydrogen. That somebody here is FAD. FAD forms FADH2, right? So can I say in this step, FAD means vitamin B2 is used. Succinate dehydrogenase will use vitamin B2, right? And you know if NAD means B3, if oxidative decarboxylation means B1, if simple decarboxylation means B6, right? All that you know. So let's see the next step. Fumarate to malate. enzyme is fumarase. I already told you it is a lyase. And then malate to oxaloacetate, the last step of TCA cycle. There is no change in number of carbons. Enzyme is malate dehydrogenase. So there's simple oxidation occurring. And here NAD comes to take hydrogen and forms NADH, right? So we are done with the detailed steps of TCA cycle. Now let's again see TCA cycle with the eagle's eye view. That is, see what is happening. But other substances are intermediate of TCA cycle, which I have explained, which I have written in the circular form. Oxaloacetate, citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate and malate. They all are intermediates of TCA cycle, but acetyl-CoA is not the intermediate of TCA cycle. Now let's see the energetics of TCA cycle from 1-acetyl-CoA. If 1-acetyl-CoA enters TCA cycle, then how many NADH obtained? 1 here at malate dehydrogenase, 1 at alpha-ketoglutarate dehydrogenase and 1 at isocitrate dehydrogenase. So 3 NADH, 1 FADH2 and 1 ATP or GTP here. So total how many ATPs? 3 NADH, 3 into 2.5 is 7.5. 1 FADH2 will give 1.5 and 1 ATP or GTP gives 1 ATP. So this is total 10 ATPs per acetyl-CoA. So these 10 ATPs are from 1-acetyl-CoA. Now let's count from 1-glucose and that will be complete breakdown of glucose. Now we are going to count. So 1-glucose will be converted to 2-pyruvate. 2-pyruvate via link reaction will make 2-acetyl-CoA and these 2-acetyl-CoA will go into TCA cycle, right? So energetics is in glycolysis, glucose to 2-pyruvate glycolysis, we get 7 ATPs because it is aerobic glycolysis, end product pyruvate. Then in link reaction, I told you 2 NADH are formed. So 2 NADH will give 5 ATPs and when 2-acetyl-CoA will go into TCA cycle, then they will give 20 ATPs. So this total is 32 ATPs from complete breakdown or complete oxidation of glucose. That is 30 ATP per glucose, complete breakdown or complete oxidation, right? So if they ask you that aerobic glycolysis gives how many, you will say 7. But if they ask you complete oxidation of glucose, then you will say 32. So be careful what they are asking. Now compare these two enzymes. One is alpha-ketoglutarate dehydrogenase of TCA cycle. Other is pyruvate dehydrogenase PDH complex, that is the link reaction enzyme. Both these enzymes are multi-enzyme complex. Both these enzymes require same 5 coenzymes, I already told you. But the difference is this point that alpha-ketoglutarate dehydrogenase is not regulated by phosphorylation and dephosphorylation, but PDH complex enzyme is regulated by phosphorylation and dephosphorylation. That is, insulin in fed state will cause dephosphorylation and will activate link reaction. Link reaction is activated in fed state. But on the other hand, in fasting state, link reaction will be inhibited by glucagon, which is doing phosphorylation, right? Now TCA cycle occur in aerobic state. We all know it occurs in aerobic state. But any enzyme of TCA cycle does not require oxygen. No enzyme of TCA requires oxygen. Then my question is, why TCA cannot occur in anaerobic situation? See the reason. Let's say there is a situation that oxygen is not there. When oxygen is not there, then TCA will occur because no enzyme of TCA requires oxygen. So TCA will occur. That is, first few cycles of TCA will be occurring. Now when TCA will occur, then lots of NADH and FADH2 will be produced. This NADH FADH2 has to go into ETC for giving ATP. But we have taken the situation oxygen is not there, so ETC cannot occur. So NADH and FADH2 will accumulate. NADH, FADH2 accumulate. Or we can say NAD and FAD is depleted. That is, all the NAD and FAD of the cell is converted to NADH and FADH2. So now NAD FAD depleted means TCA will stop, right? So this is the reason that TCA cannot occur in anaerobic condition. Now let's see the rate limiting steps in TCA cycle. See, I said rate limiting steps. Usually in some pathway cycle there is only one rate limiting enzyme or step. But in case of TCA cycle, there are 3 enzymes which are rate limiting, 3 steps which are rate limiting. That is a very special thing here. Actually, in different situations, the different enzymes can be rate limiting, and they are citrate synthase, alpha ketoglutarate dehydrogenase, and isocitrate dehydrogenase. These three enzymes can be rate limiting. Let's say in some situations, citrate synthase is rate limiting. In another situation, alpha ketoglutarate dehydrogenase is rate limiting. In another situation, isocitrate dehydrogenase is rate limiting. That body has to decide depending upon various complex conditions, right? And now, irreversible steps of TCA cycle. There are two irreversible. One is citrate synthase and another is alpha ketoglutarate dehydrogenase. If both are given, then go for citrate synthase, right? Now tell me, there are how many dehydrogenases in TCA cycle, and there are how many kinases in TCA cycle? Dehydrogenases are 4, that is alpha ketoglutarate dehydrogenase, then isocitrate dehydrogenase, then succinate dehydrogenase, and the last one is malate dehydrogenase. These 4 dehydrogenases are there. Out of these, first 2 which I have written, they are doing oxidative decarboxylation, but the other 2 are only doing oxidation. And you know, in alpha ketoglutarate dehydrogenase and isocitrate dehydrogenase, NADH is formed. Succinate dehydrogenase will form FADH2, and malate dehydrogenase is also forming NADH, right? Now, there is only one kinase, that is succinate thiokinase in TCA cycle, which is the only step of substrate level phosphorylation. Now we have to see the carbon dioxide which are released in TCA cycle. They are from oxaloacetate or acetyl-CoA. The carbon dioxide in TCA are derived from the carbons of oxaloacetate. If simply question is asked, then you go for oxaloacetate. If oxaloacetate not given in the option, then you mark acetyl-CoA, right? Now, I am going to tell you the detail of this thing also. If you don't understand the detail, then you should cram only this much, right? But once in the video, I will be taking this detail. Watch it once. If you understand, this is okay, go with the concept. Otherwise, cram this, okay? So, let's see the detail. What happens actually, you know, oxaloacetate is having 4 carbons. It will join with acetyl-CoA, which is 2 carbons, and the cycle will occur, many reactions will occur. There is removal of 2 carbon dioxide, and this is first cycle which I'm talking. In the end, you know, again oxaloacetate is formed, and in second cycle, again 2 carbon dioxide are removed, right? So let's say we are doing this as we are carrying TCA reactions in lab in vitro conditions, or in the lab we are having it, so we can give colors to the carbons, right? So let's say we give red color to the carbons of oxaloacetate. Let's give black color to the carbons of acetyl-CoA. Now, this carbon dioxide which is released is red in color. The oxaloacetate which we have obtained at the end of first cycle, that oxaloacetate has 2 carbons red and 2 carbons black, okay? And in the next cycle, the carbon dioxide which are removed are black in color, okay? So, if we are talking of only one cycle, simply if we are talking of one cycle, then the carbon dioxide are from the carbons of oxaloacetate, right? But if we talk of a previous cycle also, if we are talking of this carbon dioxide, then this carbon dioxide is coming from previous carbons of acetyl-CoA, right? So it depends on the question what they are asking. But if you are clear in this detail, then be it any question, you will be able to answer, okay? So whatever question you will be able to answer, just go with the concept. I revise once that the carbon dioxide is coming from oxaloacetate from that particular cycle for which we are talking. But these two carbons which are coming in the form of carbon dioxide, these carbons are from the previous acetyl-CoA, right? So this way you can solve the questions, right? So this is it with this video. Thanks for watching.